Francois, as you can see from his slide here, he's going to be talking about the antibacterial effects of biodegradable material inspired by insect wings. So
Exactly.
biomimicry is one of my favorite topics. I can't wait to hear about more.
Perfect. Thank you very much. So thanks, Andrew, for the introduction. So my name is François, and I'm a master's student in the Department of Food Science. And today I will talk about my research that aims at evaluating the antibacterial effect of a novel as well as eco-friendly material that is inspired by insect wing and that can be used as a food packaging material. So if we started with a piece brief of background on these insect wing inspired material, so nanopillars, which are cone-like structures with dimension of 1/1,000,000th of a centimeters, were found on insect wings such as cicada and dragonfly's wing and are acting as natural defense mechanism for the insect. So when bacteria deposit on the wing, as we see here in the picture, the bacterial cell membrane gets stretched and eventually causes permanent deformation and rupture to the membrane, which will lead to cell death with a mechanism that is really similar to pressing a thumbtack into a balloon. So previous study demonstrated that contact of only 3 minutes between the wing and bacterial cell is required to observe rupture and death of more than 90% of bacterial cell presence at the surface of wing. A group of researchers of the department and I have created a material made of plant derivative component with the same characteristic and topography of what was observed on this insect wing in order to potentially elicit this similar bactericidal effect. So the project has been broken down into 3 objectives. So the first objective is to recreate this material with the same nano texture found on the insect wing. The second one will be to evaluate if the material is indeed recreated and has the same antibacterial effect as we found on the insect wing. And the last one will be to see if it could potentially kill bacteria at the surface of fresh food product to potentially decrease spoilage. So the rational and potential outcome behind the development of this material is first the reduction of utilization of plastic material in the food industry and eventually reduction of presence of plastic in the landfills. And the second potential improvement would be the reduction of any addition of chemical preservative in the food product, which increase the clean labelness of the product, which drives consumers' interest. So now if we talk about objective 1, so the fabrication of this material, so the production of this new material is mostly made with cellulose that's been extracted from cotton product. An acid-based treatment is applied on old clothes, laundry, or sheet made of cotton in order to extract the cellulose, which is the long glucose chain found in plant cell wall. And from there, with a filtration and deposition treatment, we can obtain the thin cellulose acetate similar to saran wrap by being as flexible, as transparent, but this time being biodegradable. So in order to imitate the nanopillars found on the surface of the insect wing, commercially bought crystal of cellulose that will be termed cellulose nanocrystal, mentioned as CNCs, with a shape and structure similar to what was found on the insect wings, the nanopillars. These CNCs are deposited on the cellulose acetate film that we mentioned. So we have a nice plastic type film coated with small crystal potentially capable of killing bacterial cells. For the experimentation, 2 samples were used. So we had 3% CNC and 10% CNC film, with a second one with 3 times more CNC compared to the first one. And we also had a third sample, which is a control one with 0% CNC. So theoretically, an increase in the CNC concentration should increase the antibacterial effect, which is what we will see in the next slide. So to confirm that our newly designed material has the same properties as the bacteria killing wing, viability of different bacterial cells after contact with our material was to be evaluated. 2 bacteria found in the gut microbiome were used. So we start with Escherichia coli, a gram-negative bacteria that is known to be less resistant to external stresses because of the thin peptidoglycan layer. So such as, for example, contact with pillar will be less resistant. And the second bacteria, which is Listeria monocytogenes, part of the gram-positive bacteria, known to be more resistant to stresses such as pillar due to the thicker peptidoglycan layer. So both of these bacteria are found commonly in the gastrointestinal tract of animal and are a major foodborne issue and can cause issues in humans such as diarrhea, digestive problems, vomiting, and could be a major problem for immunocompromised person, pregnant women, as well as children. And these 2 bacteria are cause of many food recall in the recent years, such as E. coli that will be most commonly found in beef, lettuce, fruit and in North America, but also anywhere else in the world. And the presence of Listeria monocytogenes has made a thousand of recalls, especially for deli meat, egg, and any other ready-to-eat product. So different cell viability tests were performed and a result were concluded. So after 3 minutes of contact between bacterial cell and the food packaging material, 90% of these bacteria were killed on the surface and recovered after the cell count was calculated before and after. So to make it even more visual, we have here a live dead staining image. So the bacteria before contact, we have E. coli on the left side and Listeria monocytogenes on the right side. We see that green bacteria will be the live one and red one will be the dead one. So we see that after contact on the left side, both on the left side and on the right side, we have a much higher proportion of red cell compared to the initial green pictures. So with positive result and significant antibacterial effect, we were then able to pass to the second part of the project, which consists of the evaluation of the potential of the utilization of this material as a food packaging material, especially for a raw meat product. So the rational and thinking behind that is that if it can kill bacteria in a solution, it could also kill spoilage and foodborne pathogen bacteria at the surface of meat product, therefore kind of delaying spoilage and foodborne pathogen bacteria, delaying spoilage and extending shelf life. The targeted food product are meat product. Why meat? Because they're amongst the most lost food product with 20% of the globally meat produced. So we see that here we have a normal piece of meat, and after incubation with the material for a period of 14 days, we have a much lower proportion of bacterial cell at the surface. For example, compared to a control meat that will have a much higher proportion compared to the initial at 14 days. So we see that we have a clear reduction in the bacterial load. However, a lot of further testing, such as sensory evaluation, shelf life testing, and many more will be required to confirm the potential shelf life extension properties of the material. So in conclusion, we are willing to bring on the market this novel and value-added food packaging material that differentiate from the other type of packaging because it is both biodegradable and antimicrobial. And by that, we are obviously hoping to be part of the solution to solve the striking problem of food and plastic waste. Thank you very much.